I pulled in.
Welcome everybody. Welcome to the last colloquium of our spring semester, sponsored by the Genetic Engineering and Society Center. Thank you for being here. We always start with just events that are happening in the GES world over the next week or so. Anything that we should know about? Okay, I'll mention one thing. Katie Barnhill-Dilling, who is a one-year IGERT fellow, is defending her thesis next Thursday at 10:30 in Jordan 1214. I think that was listed on the announcement that Patty sent out. And much of her work has also focused on the Chestnut case. So if today's topic interests you, you might want to come and hear the public part of her defense next week. Yeah, Brett.
Friday, all day, is the Keck Behavioral Biology Student Postdoc Symposium, and some of our GES people are speaking in that. So if you want more information on that, let me know.
And Zach, before you take another bite,
sure.
Do you want to say something about the GES Symposium in June? Just to remind people?
I mean, I guess we say that every time. But
Okay.
But yeah, there's a
But since this is the last book,
oh yeah,
once you come in person.
so there's a symposium coming up June. Oh my gosh, Fred, what is
4th and 5th.
4th and 5th. Thank you. So it's basically a capstone for the IGERT, for the PhD student IGERT program, and it will be focused a lot on, the next generation training of students, graduate students in this type of interdisciplinary space. So, you'll be getting a lot more information about that. We're meeting about this afterwards for an hour, so you'll be getting
So you could come to another meeting if you like. Yeah. Jen?
Destiny found out this morning that she got an undergraduate research grant for the summer.
Yay!
And what did you win, Jen?
Oh, wow. I won a poster session at BASF last Friday.
Any other news or announcements? Great. Okay, so for today we have Andy Newhouse with us. I'm really excited to have him speaking to us today. He is a PhD student in the laboratory of Bill Powell at SUNY ESF. That's the laboratory that's developed the transgenic chestnuts, Transgenic American chestnut tree for potential restoration. That's a project that I've been studying for a couple of years as a social scientist, along with Jessica and Katie, Andy Bender, Louis Rivers, and Dalton has just come on that project as well. It's a fascinating case study in terms of thinking about the application of biotechnology to a really different kind of vision for a plant to go out in the environment in terms of purpose, for ecological restoration, and also in terms of how this GMO lives in the world, in that it's designed to spread and persist in the environment instead of live one season as a crop and be plowed under and harvested. So it's a really interesting case from many different perspectives. The great thing about having Andy with us is that he's been in that laboratory for a number of years and has worked at the molecular level, the organismal level. That's him next to the tree in the bucket. And he also has been working really hard on permits and applications for regulatory approval or deregulation of the organism. And that perspective is really valuable to us in terms of us thinking across disciplines. Bill Powell has spoken on campus before, and I thought it would be I wanted to invite Andy to speak to us, partly because Bill has spoken here before, but also because our community has really been built on these interactions between students and faculty, and the student perspective has always been really important in GES. And so I thought it would be even more interesting for us to hear from a student in the research group rather than Bill, who gives these, Andy gives these talks a lot also, but instead of hearing Bill's roadshow, which you can see as a TED Talk, I thought it would be really great to hear from Andy. So thank you so much for coming today. I really look forward to your comments.
Well, thank you very much. I've watched several of the live streams from these colloquiums throughout the semester, so it's an honor to be here in person. Thanks. And with that introduction, I don't think I need to say too much else about myself, but I guess just kind of some of the reasoning why I started working on the regulatory aspect is that I was seeing the progress made in the lab and the progress made in other labs too. The technology is here and it's usable and it's pretty accessible to most research labs, but the I guess roadblock or transition from lab work to being able to deploy these technologies in the field for environmental or conservation purposes is right now the regulatory system. So that's kind of why I jumped into that, to hopefully make projects like this a reality. So I'll say also that the work I'm talking about, some of it's stuff I've done, but really we've had a large and very active team over the last 27 years, I think, at SUNY ESF. So with that, let's start right off with a little bit of chestnut background. As you're probably aware, if you know anything about chestnuts, these were huge and majestic trees, common throughout forests in the eastern US. They were useful for a variety of purposes. Most people are familiar with chestnuts kind of roasted on an open fire for a food, but the nuts can also be ground into a flour for baking or even used for brewing. And the wood was useful for a variety of purposes, from furniture to things like log cabins and fence posts. It was durable, rot resistant, pretty unique compared to a lot of the hardwood we have today. And then of course chestnuts were useful for a variety of wildlife, either the trees becoming habitat or actual food in the case of the nuts. All that changed, of course, with the introduction of chestnut blight around the late 1800s, early 1900s. And this was caused by a fungus introduced from probably Japan, but Asia at least. And American chestnut trees were very susceptible. And the way the fungus kills American chestnut trees is by secreting a toxin called oxalic acid. This dramatically drops the pH around the margin of these cankers here. And so that acid, the oxalic acid, is what kills the American chestnut tissue, cambial tissue under the bark, and then the fungus kind of comes through and eats the dead tissue. Now, American chestnuts are not extinct. The fungus doesn't kill the roots of the trees. And chestnuts happen to be very good at stump sprouting, or sending up new sprouts from the root collar. So that happens even if a large tree like this gets the blight, dies back, it'll send up new sprouts. And those will last for a few years to maybe a decade, and then that gets knocked back again by the blight. So they're stuck in this cycle, kind of trapped as a shrubby understory species right now. There are various options for kind of dealing with if you want to do something about chestnut blight. What are some responses? Of course, doing nothing, just letting them be is one option. But I want to remind people that that decision has consequences. If we do nothing, we'll lose chestnuts eventually. This kind of resprouting from the roots can't last indefinitely, and they're, like I said, basically functionally extinct right now. They're not really serving ecosystem functions. So doing nothing has consequences. Breeding is another option, either planting hybrids, and in some cases, if you want to attract wildlife to a hunting plot or something like that, then hybrid chestnuts might be a fine option. But as far as restoration in forests, hybrid chestnuts or relatives of the Asian chestnuts wouldn't be the best option. I'll get to back crossing in just a minute, but mutagenesis is another option that's actually been tried. Basically blasting chestnuts with radiation and growing them up and see what mutations happen to be useful. And biocontrol, either using hypovirulent forms of the fungus. So the fungus can actually be infected by a virus, which weakens the fungus, weakens the pathogen. That's one option. Using biocontrols, other bacteria and so on can reduce the kind of pathogenicity of the fungus on a kind of individual tree basis. And then, of course, genetic engineering. And with all of these, I like to kind of think about which of these involve risks. All of them, really, realistically. And the difference is that everything above the line here is not regulated, and genetic engineering is regulated, so I'll be getting into that in a bit. And so just in terms of thinking about risks associated with genetic engineering or using biotechnology for environmental restoration in general, I always like to keep in the context of what are the risks relative to traditional methods. So back crossing, I'm not going to go into a whole lot of detail, but basically starting with wrong button. Starting with an American chestnut and a Chinese chestnut, you would get an F1 hybrid. If you take that F1 hybrid and cross it back to American chestnut for several generations, you get a tree with more characteristics of American chestnut and hopefully the blight resistance of the Chinese chestnut. So this involves breeding out unwanted traits and hopefully retaining the traits of blight resistance, ending with a tree that is about 1/16 Chinese chestnut. And this has been a long-term research project of the American Chestnut Foundation. And most of you are probably pretty familiar with genetic engineering, but this is a slide we like to present to general public type audiences to kind of put genetic engineering and the scale of these changes in context. So since the chestnut, American chestnut, has about 40,000 gene pairs, we use the metaphor of a book that has about 40,000 words, so to words corresponding to the genes in the tree. So in the case of back crossing, where you have about 1/16 Chinese chestnut, if you imagine about 1/16 of the words in this book to be in Chinese, would that matter? Could you still read the book? Probably. It depends on kind of where those changes were made. In the case of genetic engineering, we're making very small and very precise changes. So this is an excerpt from Walden. It was very exciting at that season to roll in the then boundless chestnut woods of Lincoln. And instead of swapping out a couple thousand words in the case of back crossing, we're adding 2 words, or 2 genes to the tree. So the then boundless blight-resistant chestnut woods of Lincoln. We know what changes we're making. We know, in our case, we know after the fact where they were inserted, but it's a very small, very well-characterized change in contrast to breeding. And this results in a tree that's really 100% American chestnut plus 2 genes, as nothing is swapped out. The gene that we've added, that we've had the most success with at least, is called oxalate oxidase. And remember I said that the blight fungus releases this oxalic acid, and that's what kills the tree tissue? The gene that we're adding codes for an enzyme that breaks down oxalic acid. It's called oxalate oxidase. The byproducts are hydrogen peroxide and carbon dioxide, both of which are neutral or beneficial to the tree. And again, for kind of general public audiences, I say if you're not too into the chemical formulas, here's the cartoon version. So we have Buster Blight over here using his acid attack on Charlie Chestnut, but we've given Charlie Chestnut an oxalate oxidase shield and protecting him from the effects of the blight. So then Charlie and Buster can live in harmony. A key part of this is that the oxalate oxidase doesn't kill the fungus. It's not an antifungal. It doesn't have antifungal properties. It doesn't deter the fungus. It breaks down the acid that's produced by the fungus. So for one thing, that reduces chances that the pathogen can overcome this resistance. It's not a strong selective pressure that it would be by killing the fungus. And it also might have regulatory implications that I'll get into in a bit. We also have an easy assay to identify which trees are transgenic. And this gene, oxalate oxidase, the one we used came from wheat, but it's present in a wide variety of other plants also. Just about all of the cereal crops have oxalate oxidases, and even wild grasses, even unrelated things like strawberries and bananas and mosses. So the gene is already present in our food. You've probably eaten oxalate oxidase today, and it's already present in the environment. It's not a foreign compound. So I'm going to go real quickly through our whole process. Basically, I want to say that we've used agrobacterium transformation, which by today's standards is kind of old-fashioned in the genetic engineering or biotech world. But, and then we've transformed what are called somatic embryos. And then those can be grown up, well, the cells that we transform grow into embryos, which can then be induced to form whole plants. But that means these whole plants grew from a single cell that was transformed, so they're clonal, they're genetically identical. And as you can imagine for a restoration program, we don't want to distribute or plant a bunch of clonal trees. So the next step is pollination. And so we take these trees that we produced in the lab, the initial transformed lines, and we grow them in highlight growth chambers, usually indoors, until they produce pollen. We can collect that pollen and then do pollinations outside. These, any pollinations we do right now are inside our permitted plots. But then the offspring, if you remember basic biology, from the chromosome, a single copy transformation only has one of these paired chromosomes with the gene. So about half of the offspring from these crosses with a transgenic parent and a non-transgenic parent, about half of the offspring will inherit the trans gene. And they do have the same resistance as the transgenic parent, but more genetic diversity. Another benefit is that growing these tissue culture plants straight out of the lab is kind of finicky. They grow slowly. It's more difficult to get them established. After they've been established, kind of get rooted in the ground for a couple of years, they grow at about the same rates. But certainly initially, you can see that the actual nut, it's a big pocket of stored energy and nutrients. It really helps get the plant started and grows faster for the first couple of years. So that's another benefit to doing these pollinations, to distributing actual chestnuts instead of these tissue culture lab trees. In addition to the genetic engineering, or the genetic diversity is the ease of planting and distribution. So the next kind of natural question is, does it work? And yes, this is a assay that we did in the greenhouse a couple of years ago on small stems. On the far left are non-transgenic American chestnut controls, so susceptible American chestnuts, and they're completely wilted after, I think that this is about 6 weeks after we inoculated the stems in the greenhouse. You can see some new growth coming from below the inoculation site, and that's typical of how they respond in the wild, also kind of like stem sprouts. On the far right are the Chinese chestnut controls. The Chinese chestnuts are considered resistant to the blight, and these look small, but we selected them for the same stem diameters as the other types. And in this assay, about half the Chinese chestnuts wilted too, because this is a relatively heavy load of the blight fungus compared to the diameter of the trees. And then the transgenic American chestnuts are doing fine with no wilting at this point. And then another example, this is from a field assay, and these are actually seedlings. So these are the T1s that we've pollinated. And then this one inherited the transgene, and this one did not inherit the transgene, and you can see a very different response to the blight. These are full siblings from the same pollination, and I also want to mention here that this is an example you can see around the inoculation site. If you look closely, there's some orange tissue there. The fungus, the blight fungus, is still growing. It's still active. It's still colonized that wound, but it's limited from doing the same kind of damage it does on a wild-type American chestnut tree. And so this is a case of what we would call tolerance. The tree is not really resisting the fungus. It's not repelling or preventing the fungus from colonizing the wound. It's not killing the fungus, but it's tolerating an infection of the fungus with very minimal damage. And I also want to mention this is the transgenic American chestnuts look really remarkably similar to Chinese chestnuts. This is a different field inoculation. It wasn't done at the same time as these. But you can see there's damage from the wound where we did the inoculation. There's a little bit of colonization of the fungus, but it's really kind of a superficial wound, and the tree hasn't died from those inoculations. So we're really kind of recreating the situation as it exists in China, where the blight fungus is native, with these transgenic American chestnut trees. So not killing fungus, but allowing the trees to coexist with the fungus. The next natural question after does it work is it safe? And this is a really texty slide. I know you don't have to read the whole thing, but just these are some examples of kind of comparison studies or non-target studies that we've done on other organisms or other interactions in the environment. The only one I'll mention here is mycorrhizal fungi. A lot of people ask, oh, you're trying to protect this tree from a fungus, but what about the beneficial fungi, or mycorrhizae that live underground? And so we've done actually several different assays on transgenic American chestnut trees compared to other sorts of chestnuts, and we're consistently seeing the same mycorrhizal colonization on transgenic versus non-transgenic chestnuts. So that relationship is not being inhibited or affected, and should have a publication. It's being reviewed right now, but that's coming out soon. And then I'm going to really briefly go through just a couple other examples of these studies. One is tadpoles. And we put tadpoles in jars with some leaf litter, so crushed dried leaves from different types of chestnut trees, and then a couple of other trees, sugar maple and American beech, that are currently prominent in our hardwood forests around Syracuse. And real briefly, just looking at survival, this is our whole setup. And the idea is that tadpoles live in vernal pools, which are lined with deciduous leaves. So this is an interaction between deciduous trees and tadpoles. It might not be immediately intuitive. But looking at survival of tadpoles in different types of leaves, this statistical analysis is called a Cox proportional hazard model. And what you're looking at here, the 1 is kind of a reference point, and so sugar maple was set to 1. That's considered kind of the neutral, the baseline, or what's out in the woods right now. And the only thing that had an increased survival hazard compared to sugar maple was American beech. So tadpoles didn't do as well, didn't survive as well on American beech as they did on sugar maple, but all of the chestnut types, whether it's Chinese, hybrid, non-transgenic, transgenic, all the chestnut types had either the same or even reduced survival hazards, so they did better or the same compared to sugar maple. Tadpole development was also an interesting story. I don't have time to go into a whole lot of detail, but basically the transgenic and non-transgenic American chestnuts developed just a little bit faster than everything else when they didn't receive supplemental food. So it's possible that chestnut restoration in general, whether or not it's transgenic, could actually benefit tadpoles in vernal pools, but that's a complicated question. Moving on to bumblebees. Native pollinators are of course a concern with generally with the public right now. So we did these studies using what are called microcolonies and bumblebees with chestnut pollen. And we actually applied oxalate oxidase enzyme to the pollen. We can't grow trees big enough to collect enough pollen, transgenic pollen straight from transgenic trees, so we added the enzyme to the pollen for these tests. And then use these microcolonies which allow bumblebees to have some social interactions as they do in the wild, but still in kind of manageable experimental units. And we saw that in terms of survival, these are the 3 different pollen types and they're all about the same, whether it's no oxalate oxidase in the pollen or kind of standard amounts or high amounts, there's just no difference in survival. And then we looked at pollen usage over the scale of the experiment also, and no significant differences in pollen usage. And it actually looks like the standard concentration of oxalate oxidase, the bees might have used a little bit more of that pollen, if anything, but that was not a statistically significant difference over the whole time series. So consistently with all of these ecological studies we're doing, we're not seeing risks with transgenic trees, transgenic pollen, transgenic roots, whatever the case is. We've also done some nutritional analyses, which will be part of the regulatory process also, again, just a reasonable precaution with introducing something new. What you're seeing here in the green columns are 2 different types of transgenic chestnuts. These are from the pollinations that we did in our permitted plots. And then next to them is the full sibling, a non-transgenic from the same cross, or ones that didn't inherit the transgene. And so these are 2 different years of analyses. And if you look closely from within the same year, the transgenic and non-transgenic pair is very similar all the way down, no significant differences. If you look between different years, there are some differences. Or if you look, these are other non-transgenic chestnuts. If you look just from one tree to another within the same year, we see some differences. That's probably differences in maybe rainfall, in soil micronutrients, and where these things are planted. There are differences that we can pick up between chestnut trees, but not between transgenic and non-transgenic chestnuts from the same crosses. So. Then other tests that we'd like to do, really pending regulatory approval, like I said, are some larger scale, longer term ecological studies. We just can't do this in our current permit setup. I'm not getting into that now, but we have these plans to do larger scale plantings, maybe putting in some real vernal pools in with different types of, these would be different types of chestnuts, looking at how quickly they disperse, pollination distances, and so on. But like I said, this is not really possible under our current permit system. So moving on to that, currently anything we plant outside or anything we move across state lines or any pollen that goes outside has to be covered under an APHIS permit. And so these are all considered temporary confined field trials, and they're basically set up so that propagable transgenic material can't accidentally be released or into the environment. This involves a lot of paperwork. We submit reports for everything we plant, everything we harvest, everything that goes outside has to be on these permits. And then that also involves inspections from APHIS almost every year now. We do sort of have permission to let trees flower outside, but there's a whole long list of conditions that go with that. Primarily that there can't be any compatible chestnuts within a quarter mile of where the flowering trees are. So it basically eliminates the chance that anything could be accidentally pollinated. But in reality, those conditions are pretty tough to meet, and so we're doing controlled pollination. So everything's inside our plots, all pollen is bagged, the flowers are bagged at all times to prevent the pollen from getting out. So moving on to the actual regulatory process. When we want to submit these to be able to distribute them, to hopefully be able to use them for restoration, there are 3 different federal agencies who would be involved. Actually, one outside the US. In Canada, there are a couple of agencies in Canada who we would also submit to if, since the chestnut range extended into Canada, it would be likely that if in unconfined releases that the trees would end up in Canada at some point anyway, so we would be submitting to their regulatory agencies also. Then the first US agencies is the FDA, which evaluates food and feed. People and animals both eat chestnuts, so FDA is involved. They're looking at nutritional analyses, composition, things like potential allergens or toxins that would be different in the transgenic product compared to non-transgenic. And then if they are happy with what we've submitted them and they determine there are no nutritional food feed safety risks, then they come back with this response of no further questions. And technically the FDA part is voluntary, but basically everyone does this. And the EPA and other groups would be consulting with the FDA anyway, so it's not like we could hide anything from them, nor would we want to, of course. Second agency is the EPA, and EPA regulates pesticides. And since there's this interaction with a pathogen, EPA is involved to some extent. So the law that they're upholding is called FIFRA, and they define pesticides as a substance or substances intended for preventing, destroying, repelling, or mitigating any pests. But our case is really unique. Remember I said we're not killing or repelling or preventing or destroying this pathogen. So we're still in talks with the EPA to determine exactly what our regulatory pathway will look like with them. But typically what they do is establish a tolerance limit or an exemption. And in our case, since oxalate oxidase is already out there, they can't very well say we're going to regulate oxidase when it's already in wheat. So that it just kind of doesn't make logical sense. But so that it would probably look like an exemption or a tolerance exemption in our case. And then they issue what's called a registration, which you can imagine if they're reviewing a corn variety or a jug of chemical pesticides, they issue this registration and then they come back after about 5 years and kind of review it, see how it's going, and determine whether they're going to renew it or whether the developer has a newer and better product they want to register anyway. But in the case of a tree that's long-lived and you would like it to be out in the woods and breeding with wild relatives, that registration process just doesn't really make sense. So the idea of coming back to review something and then deciding whether to reissue a registration just doesn't make sense. So again, we've been talking to the regulators and kind of figuring out a reasonable path forward with that, but that has not been totally firmed up with the EPA. The USDA is a little more clear. They basically regulate sort of safety for agriculture and the environment, I guess I would say. And the trigger, the reason that they're definitely regulating us is that we've used agrobacterium transformation. Agrobacterium is considered a plant pest. So even though there aren't agrobacterium pieces, residues in the tree, the fact that we use that as part of the process triggered USDA regulation. There are, I thought that was my 5-minute warning, pretty close. There are a couple of different parts of their process, also a plant pest risk assessment where they're trying to decide whether the new product in question has any new risks, any enhanced weediness, or would present a risk to people or the environment. And then NEPA, where they're doing an environmental impact statement. As part of this, there are open comment periods also where people, they're soliciting comments from the public. It's mostly for things like, would there be risks they haven't considered? Are there parts of this, other things that they should review or question as part of the process? There are other groups that are kind of partially involved in this process also, not directly regulatory groups, but kind of consulting groups in the US, places like the Fish and Wildlife Service or the Forest Service will probably be consulted by the regulatory agencies, but they're not really decision makers exactly. Other groups, general public can weigh in and so on to move along here. But thinking more generally about other kind of regulatory considerations, we're looking at probably a couple of years after we submit the petitions, which will hopefully be pretty soon. We don't have a real firm timeline. But there's been a lot of variability. There have been a few examples recently where a new product has gone through in a year or less, and others that have been in the process for 10 or more years and are still kind of tied up. We don't really have a template to work from here, but really our petition will probably become a template for other projects in the future. So, also regulatory permission basically covers the product that we're submitting, but also all offspring, so that makes sense for kind of breeding and distribution. One kind of trade-off that we're constantly thinking about is balancing privacy and security. You're probably aware of anti-GMO groups being involved in things like arson, damage to labs and field plots. But we also want to be transparent. We want to involve the public. We want people to see what we're doing. We don't want to work entirely behind closed doors, but we need to kind of balance those two things. We don't want to publish the address of our field plots, but we do want to give tours to anyone who's genuinely interested in our stuff. So that's kind of a tricky balance. And both tree research and dealing with the federal government require a lot of patience. So dealing with both at the same time is really this is a project for future generations. These are my kids hugging some chestnut trees, of course, and hoping that they will be able to see chestnut trees in the woods. And very briefly, I just talk a little bit about the Chestnut Foundation's plans for distribution. And like I said, we want to encourage or supply appropriate genetic diversity before we actually distribute these things. We've been working closely with the Chestnut Foundation, and they will actually probably be managing more of this distribution restoration aspect. So they've done some modeling and come up with basically, we're hoping to breed our transgenic chestnuts with something like 500 different non-transgenic chestnuts from different parts of the range, and then cross those for multiple generations before there's really kind of an optimal level of genetic diversity to support healthy populations moving forward. I think I just need to skip phytophthora. If you have questions about that, feel free to ask. Overall, responses to our project have been very positive. A lot of support from the Chestnut Foundation, from articles, publications, presentations. The majority response that we see when we give presentations to the public, first question is, when can I get a tree to plant in my yard? And that's exciting. I want to plant a tree in my yard too. So a lot of our response has been positive. There are a few kind of very dedicated anti-GMO groups that seem a lot more interested in just blocking things than actually having a productive conversation. And I'll mention that some of those things are kind of conflated with other issues. So I saw an article recently that was talking about chestnut and then said, oh, the Bt genes for insect resistance are going to mess with the ecology. There's no Bt in chestnuts. That's just a separate issue. That's worth talking about, but it's not in chestnuts. So that's part of what's going on. Thinking about chestnut diversity in terms of restoration, this was an interesting paper that showed a lot higher levels of diversity in the southern part of the chestnut range. Kind of, you can imagine a glacial line around here, and then as chestnuts move north, they're less diverse. And then research that you're probably familiar with from Jessica, looking at modeling climate change and potential restoration locations, future chestnut habitat. It's also something the Chestnut Foundation is considering. And out of time for the really interesting questions, kind of post-scientific questions, I guess. But thinking about who starts the research, who benefits from the research, thinking about the actual restoration, I don't really think that should be the scientists who have produced this tree that are actually managing, pushing for the restoration. The Chestnut Foundation, in our case, has been really pushing for that, and that's the public group. That's the group of people who are enthusiastic about chestnut trees pushing for restoration, rather than the scientists who have directly produced the product. So I'm excited for this, the upcoming workshop, thinking about decision-making surrounding these trees, and my kid's sitting in an ash tree, so that's another kind of idea of thinking about long-term potential for biotech for restoration. I hope that, my view is that I hope that we can do something about the environmental problems that humans have caused, and I think biotechnology is one way to do that, should at least be considered. So, with that, I'll take questions.
I've got a question about the restoration process.
Yeah.
And so chestnuts are going to face lots of other challenges if they go back out into this enormous restoration project, which would be really remarkable in lots of ways if it were to occur. And that's true whether or not we're talking about the back-crossed Asian chestnuts or the GE chestnut. And I'm thinking of things like changing forest species that prey on or fail to prey on the nuts, the distribution of the seed. All sorts of things are going to be different from what they were in the 19th century when chestnuts were at their peak. So here's the kind of wrinkle. Since you know conditions are going to be different in the future, why not add more genes to these chestnuts to make them adaptable to those conditions, to help them succeed in the conditions that Jessica plotted?
Yeah. So more transgenes.
That's the difference between your chestnut and the back-crossed Asian chestnut.
Right.
Because you can actually think about the future rather than just the past.
Right. Yeah, that's a good question. And personally, I like the idea of more diversity. And whether that's combining our program with the back-cross breeding program, introduces some genes from Chinese chestnut. So there might be multiple kind of mechanisms of resistance to the blight for one thing, but also just kind of more diversity, more potential for responding to other challenges, other threats, other climatic conditions potentially, like you said. But as far as more transgenes for more resistance to known threats like Phytophthora, or kind of less well-characterized threats like climate change, that's a possibility. But at this point, we really need to kind of figure out the regulatory process and start with the kind of most well-characterized threat of blight. But also things like Phytophthora resistance is another disease prominent in the southern part of the chestnut range. There are Chinese chestnuts that are pretty well, they have good resistance to Phytophthora. So that would be one way that crossing transgenic chestnuts with some of the back-cross chestnuts or other hybrids that have resistance to Phytophthora from the Chinese chestnut, that might be able to get kind of the best of both worlds without going through another regulatory process or another transgene. But certainly incorporating diversity from different parts of the range would be a big part of that, responding to other threats. Does that answer your question? Yeah.
Yeah, you brought up a couple different potential ecological risks that you sort of seem to anticipate. To what extent was that sort of mass list of potential ecological concerns generated by your team versus agencies from the government, or generated maybe by the public in talks that you had, and how did you sort of whittle down? How did you create the big list and then whittle down what you were actually gonna go forth and do?
Good question. Some of it is regulatory, trying to answer questions, but as we were kind of having early talks with the regulatory agencies, they don't give us a checklist of these are some things you need to answer. They, especially talking to the USDA, they said, well, submit everything you think we need to know to make a decision. Great, okay. So we tried to do kind of representative studies in a variety of different types of relationships. Some of them were things that were kind of commonly brought up, things like pollinators. Everyone's concerned about bees, so that's a common one. We've done other insect feeding stuff, and also tried to think about possible realistic risks. So people say, interactions with other microbes, especially interactions with other fungi, since that's kind of a question of the blight fungus. What about beneficial fungi, like I said, with the mycorrhizae? So tried to, several of the things you said, some of them kind of fit well into regulatory questions, some of them kind of answer common public perceptions, some of them kind of answer what we thought were most likely risk scenarios, so a variety.
So about 5 slides back, you had a study that was done in 2017 with the map and variation.
Yes.
And in looking at Canada, it seemed like there was a certain greater degree there, yes.
That's
And
a really
yeah,
interesting one.
and so could you explain that that larger share of variability that far north
Yep.
versus what we see here in North Carolina, even in our west?
Sure.
And is
Yeah.
that being brought into play in any way?
Yep. That's really, I'm glad you asked. I was hoping someone would ask that. But in this paper that just came out a couple of years ago, they said, there's this pretty much linear gradient of diversity from far south to north, except for this population in Ontario. And what they proposed in this paper was that indigenous groups, Native Americans, as they moved north, brought chestnuts with them. So it kind of bypassed the traditional tree migration routes and transported chestnuts directly, probably from farther south, to Canada. So that's, I don't know that anyone can prove that, but that seems like the most likely explanation and what they suggested in the paper.
I'm interested in the breeding to increase the genetic diversity before you release the transgenics. Do you, you might not know, but do you have a sense of what the effective population size was prior to the blight, and then what that breeding scheme will produce?
Yeah, good question about breeding and population size. The estimates are that pre-blight there was something like 4 billion American chestnut trees. So just thinking about that scale, we're never going to, every nursery is not going to produce 4 billion trees, but the modeling, which direction was it? The modeling that Jared at the Chestnut Foundation did was looking at an effective population size. That was incorporated in his models. This 500 different chestnut trees and then 3 generations of breeding with unrelated trees was intended to produce a healthy population range-wide. So I don't know the numbers on that, but is that
Well, I mean more genetic effective population, which is not an actual number of
So
trees,
sort of
but
genetic
like,
diversity within the range. That I don't know exactly. That was partly described in this paper, but I guess as I understand it, there's more genetic diversity of chestnuts in Asia and Europe, and that's kind of the center of origin for the genus. And then the southern part of the chestnut range, like described in this paper, is the center of diversity in North America. There are other members of the chestnut genus called chinkapins in Ozarks and also kind of farther southeast. So again, there's definitely more diversity in the southern part of the range, but I don't know.
that's fine. Yeah.
So the 500 number comes from a model
Yes,
that was run by the Chestnut Foundation.
that's right. So it's in the hundreds. There have been a couple of different options, 300 or 500 unrelated wild type American chestnuts from different parts of the range. But then he also, there's also talk of incorporating sources of resistance from the Chinese chestnut lines that the Chestnut Foundation has used in the backcrops program. And so possibly kind of stacking mechanisms of resistance, like I said, but that would probably be separate from kind of a pure American track. So give people sort of options for that.
So when you were introduced, there was something important that was left out.
What's that?
And because before you started working, the chestnut, restoration of the chestnut depended on 2 people who could look at a tree and tell you the extent of resistance that that tree had. There were only 2 people in the world who had the confidence to do that. One in Connecticut and one in Meadowview.
Mm-hmm.
But the problem was that they didn't agree. And so the future of restoration of the American chestnut rested on this shaky conflict between what was resistant and how resistant it was and what it was, until you developed the leaf assay, which allowed you to quickly and quantitatively determine the extent of resistance.
Yep.
For which you should be very proud.
Thanks. Yeah, I took the leaf assay slides out, but we developed an assay to, especially as we were producing a number of different transgenic lines, we tested individual genes from Chinese chestnut to use as transgenes in American chestnut to see how much resistance those individual genes conferred. And so we had literally hundreds of trees, of little trees in our growth chambers that we didn't really want to plant outside and then wait 5 years before we could do traditional inoculation. So we developed this leaf assay to quickly screen trees when they were this big rather than waiting 5 years outside. And that's been, there have been questions since then about exactly how quantitative it is, but it does a good job of kind of broad categorization of very resistant versus very susceptible.
Just going back to your risk assessment targets that you went over, I guess one thing, you talk about ecological risk assessment, and I guess that long-term ecological research slide really gets at my question, how to think about it. But you're talking about potentially transforming the entire, eastern forest over. I mean, that's
take
lifetimes. It would take a very long. And that's actually my question. I mean, do you consider that if you're going to transform eastern forests over a very long period of time? Do you consider that part of your responsibility to think about what that means, or is that so far in the future that it's just not really part of your worries, or do the regulatory authorities even bring that up? Conversation.
No, that's definitely a question, is what will this look like? What will this mean for the future of the species, for the future of other threatened species? And I guess since you asked kind of what my perspective on it is, I would say as a scientist, my role would be to, hopefully create a chestnut tree that could be used for restoration. And if broader groups like the Chestnut Foundation and other interested individuals want to use it, great, I hope that happens, but I don't see it as my personal responsibility to go and Johnny Appleseed chestnuts through the forest. Does that distinction make sense?
So, one thing that's not really considered is that the forests are not, or have already been transformed, and they've been transformed several times. There was a time not so long ago when chestnut was a rare species, when it was not abundant. And then it became more abundant. Some argue because of indirect management by Native American populations. And then it was transformed again by the blight to reduce the chestnut. So it's gone from being low in abundance to being high in abundance, to being low in abundance within, let's say, maybe 1,000 years. So it's not a question of transforming forest, it's a question of exactly what is going to be changed. But they're not the forests they were, and maybe not the forests they ever were.
Right.
Because they're changing all the time.
It's just kind of an intrinsic restoration question of what are you restoring to, what are your goals, and then those are all tied to values and so on. Really interesting questions.
There isn't a clear answer when the forests were in some stable, sustainable, long-term state. They just, these forests aren't doing that.
Right.
So you, 2 questions. When did you start engagement with the regulatory agencies? And in one of your slides, you just made a glancing comment about Canada.
Yep.
And I'm interested in what your thoughts are about the the beneficial aspects of potentially working with two regulatory agencies at the same time to maximize societal benefit rather than, you know, project benefit.
Yeah, as far as when we started, as soon as we had, actually before we had chestnut trees that were transformed to be tested outside, we worked with, before I was even in the lab, they worked with poplar as kind of a model, did some outdoor tests with poplar under APHIS permits. So that was when we started engaging, I guess, with USDA, it was just to get permits. And then as we started working with chestnuts, as that became more successful, more realistic, then kind of starting with the permits, that kind of transitioned to conversations about what would regulatory, kind of deregulatory actions look like. That was probably 10 years ago that we started kind of talking seriously about that. But that was before we really had good, solidly resistant American chestnuts that, tolerant, I should say, not resistant, but you know, so that's...
And what year was that? How many years ago would you say you got that strain?
That tree?
Yeah, that tree.
Probably 5 years ago.
So you started a conversation 10 years ago and you got the
tree. Even 10 years ago, we were seeing progress. So we've had several different kind of generations of trees with the oxalate oxidase transgene, starting from almost no difference, but the canker looks a little bit different, but it's not really resistant at all, to, oh, this tree is kind of intermediate between American chestnut and Chinese chestnut. We're making progress. And then changing things about the promoter that controls the transgene and so on, kind of was incremental progress over the years. But so that kind of, as we were kind of transitioning to talk more about the regulatory.
And have you had formal engagement with Canada?
We have, yes, not submitted anything, but we have talked to the regulatory agencies there. And we've been reminded several times that the US and Canadian and Mexican, but that doesn't matter for chestnut, but that the regulatory agencies talk to each other.
Yep.
You know, so that wouldn't be a surprise to anyone. And they just said, especially for Canada, they would be reviewing similar types of data to the U.S. agencies, so we would hope to submit around the same time. Yes, it's in progress. Yep.
I don't know, you said it, do you have any plans to use some of the newer techniques, so at least this week, say you were to use CRISPR, you wouldn't have to deal with
CRISPR, right, yeah, that's a great question. We are looking at it. There's one grad student in our lab who is kind of starting to look at that, and as we were first talking about it, or as it was becoming more popular and easier, it seems like it's really easy to use CRISPR to make small changes in a genome, but at least for the oxalate oxidase gene that we're talking about, inserting a relatively good sized large new gene is more complicated with CRISPR. And so we're looking at things like, okay, there's a native gene in American chestnut that has something like 80% amino acid similarity to oxalate oxidase, but currently doesn't perform that enzymatic function. So if we could tweak that gene to act as an oxalate oxidase, then that might be right. That might be an application of a newer technology that would kind of sidestep some of the regulatory questions. But at this point, that wouldn't necessarily be quicker or easier than proceeding with the regulatory.
I don't recall an experiment where Chinese chestnut was assayed for oxalic oxidase activity. Do you know if that's been done?
We've looked at that, and so the question is, does Chinese chestnut have oxalate oxidase activity? And really specifically, the answer is no. It doesn't have an oxalate oxidase enzyme, but it has another pathway that does some of the same thing. It breaks down oxalic acid through what seems to be like a 4-step enzymatic pathway. So we've actually screened leaves, taken a leaf disc, and soaked it in oxalic acid to see how much damage oxalic acid does. And American chestnut is very susceptible, it turns brown from oxalic acid. Chinese chestnut tolerates it better. It doesn't turn as brown, or less of it turns brown. So there is a pathway by which it's somewhat protected from oxalic acid, but not the same pathway. Does that answer your question?
Yeah.
Okay.
I saw a couple of times on your discussion slide you flipped past a list that included ohia
Yeah.
Which has been interesting to me for a long time because it involves a lot of questions of other endangered species that depend on that tree.
Yeah.
In your discussions with EPA about the chestnuts, have you explicitly talked about interactions with species that have endangered species protection that they're concerned about?
That's a good question, and there would be similar questions with chestnut. There are threatened species that relied on chestnuts, maybe more that we don't even know about. But yeah, that's a great one. The ohia tree is native to Hawaii, and it's being threatened just in the last, 6 or 7 years by an introduced fungus. And I brought it up to say that Hawaiian researchers have gotten in touch with us to kind of talk about possibilities for restoration. They're not starting a transgenic ohia program, not yet at least, but just wanting to talk to the Chestnut Project as an example of using biotechnology for a restoration program. But then as far as your question about benefits to other endangered organisms, at least the USDA regulatory process is not a risk benefit. It's not, they're not weighing benefits versus risk. It's, does the product present new risks? And EPA might be a little bit of a different question as far as potentially protecting endangered species, but that's, the regulatory process really focuses on the product and not kind of potential benefits, implications, things like that.
So somewhat related to that,
Sure.
Zach's question about, landscape level.
Yeah.
Try to keep thinking of, okay, would the ecology be better? I mean, in terms of ecological services and all of that, water be clearer?
Yeah.
Do you have any way of estimating that, or are we just doing this because, we like the chestnut?
There have been models of that. I don't know offhand, I don't remember specifics offhand, but people are looking at what are implications of chestnut restoration. And that's not really a genetic engineering question specifically,
No.
but that would apply to backcross or if there are other treatments for blight, something like that. So
The whole
definitely
idea of
interesting.
restoration, would we have a more balanced ecosystem or anything like that? So
Right.
people are looking at that to
Yeah.
see that.
I don't. We're not specifically,
Yeah.
but other people are.
A couple questions. So some of us are involved in a project with mice and potentially altering sex ratios for biodiversity. And so the first one is related to, you mentioned, you've sort of several iterations and it's getting better. Is that complicated? The risk assessment or regulatory process is basically the organism or the technology is changing while you're doing that. And then probably somewhat related, you'd also mentioned, folks who have concerns that are maybe ill-informed or conflated, and if you have lessons learned about dealing with that.
Good question.
Yeah, not
So
completely.
first, as far as kind of iterations of the product, both USDA and EPA have said if you have data from kind of earlier versions of the product, especially in our case, since it's the same gene, it's the same enzyme present in the trees, absolutely present that data. They would call that bridging data to, they could evaluate, okay, does the presence of the transgene in the tree make a difference, even if it was controlled by a different promoter, for example, in our case. So yes, that's still relevant. That's still part of our applications or petitions, even though we're not submitting those older events for regulatory consideration. They're still relevant. And then as far as dealing with opposition, it's, like I said, really it's been minimal, and in fact I think the better example is cases where people are opposed to genetic engineering. Maybe they don't like the idea of big business controlling food and pesticide usage and some of these kind of common anti-GMO themes, but they'll say, in the case of chestnut, that seems okay. People will make an exception for an environmental restoration product as opposed to maybe a row crop that's produced for profit or insect resistance or something like that. So we're always asking, if there are negative articles or something like that, we try to offer, hey, if you want to talk about this, we'll talk. We want to. We're not hiding anything. We want to be transparent. And I know other people are pushing even harder for the transparency. I think mice was an example, yeah. Does that answer your question at all?
Yeah.
Okay.
I just want to ask, anybody who hasn't asked a question, want to have an opportunity before we close? Yes.
Your response to John's question sort of touched on it. Are there any sort of commercialization feelers out there for the chestnut? I mean, it is a very hardy hardwood for construction and, like you said, furniture.
Yeah, commercialization. Good question, and that's not really something that we're pushing or hoping for. You know, as researchers, especially at the College of Environmental Science and Forestry, we want to put this out there for restoration, or for potential restoration. But we are not patenting anything. We want other people to be able to breed these. So as soon as we have regulatory authority, we hand them out, and people can do with them what they want. And we hope that nurseries start producing them to kind of ramp up numbers for restoration, for private plantings, for you know, industrial, not industrial, but potentially agricultural use. So yes, there's potential applications there, but that's not really stuff that we're pushing for. We would like to see that by whoever's interested after regulatory approval.
Well, there has been some interest in commercialization. The American Chestnut Foundation, not too long ago, had the idea that restoring the chestnut was going to be a way to put their grandchildren through college. That they were going to improve these trees and then they would sell them. So they have moderated this position somewhat, but not so long ago that was the idea by many people in the American Chestnut Foundation.
Good, well please join me in thanking Andy. And if there are any students who would like to join